Before starting, like, you know, so I want you to know something that's extremely important when you approach mole. Usually mole, it's about chemical reactions. So when you start mole, usually it's about a chemical reaction that is then another step, then step, and then another step. Okay, so it's a sequence of steps. Okay, and one of these steps is the rate limiting enzyme. So this step, for instance, is the rate limiting enzyme. It determines the rate of the entire reaction. Okay, now whenever this enzyme is defective, whenever it's defected, you will get a disease 100%. This disease has unique symptoms, okay, that will
الو
okay, I'm sorry.
بلخي وين؟ هي
طيب يلا. So we have in mole in general, you have a rate limiting enzyme which determines the rate of the entire reaction. If this enzyme questions, they usually ask about the rate limiting enzyme involved in the syndrome, or they usually ask about the thing that accumulates in the syndrome. Okay, so I want you to keep all these things in mind, and I want you to know that anything that affects the rate limiting enzyme, so if you had, for instance, I'll give you glycolysis, for example, its rate limiting enzyme is PFK. Okay, so if you had a lot of ATP in the cytoplasm, if you had a lot of ATP in the cytoplasm, that means the cell is in a high energy state. So that's why ATP inhibits glycolysis by inhibiting the rate limiting enzyme. So usually students, what they do is they memorize, oh, ATP inhibits this enzyme, ADP stimulates it, كذا 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 كذا. So after some time, or after 30 lectures, this will get extremely confusing. So you need to have that understanding how each chemical is related to the enzyme, so that you can derive the effect rather than memorize it. And I think that's a very important distinction. And you should 100% know each syndrome with its unique symptoms and the enzyme defective. Okay? طيب. Let's start with this lecture. Previously, we've talked about how different genes are expressed and what are the factors that affect the gene expression. Okay? And that we call that epigenetic inheritance. Now, let's say we had. Hala.
Can you please share the screen?
Oh, I'm not. Hello. صار لي ساعة برسم المشكلة. Okay, this is what I was drawing. طيب. So you had, I was telling you, it's a series of steps with an enzyme and how type. Well, I'll talk about it later if you text me or hi, because I don't want to take much of your time. Okay. And I've told you some of the questions that they really love to ask about in that lecture. I will do the same for this one. So now we're talking about how different proteins are moved after transcription or during transcription, after translation or during translation. Okay, so let's start. So the destination of a protein after translation is either to the mitochondria or to the rough endoplasmic reticulum or to the lysosome or to the peroxisome or to the cytoplasm or outside of the cell. So there are different destinations. Okay, and you need to know how does this happen? How does it go to the lysosome and not to the nucleus? How does it go to the mitochondria and not to the outside of the cell? and we'll talk more about this. Okay, now usually Professor Abdul Jabbar loves to put these questions at the beginning of the lecture, which are 100% not coming. So you do not need to know what diabetes insipidus is, or how it causes, and what's the mechanism of action, or how does it work. Okay, but I think that's a good thing, actually, that you start orienting yourself with these questions, because this is what they like to ask about in step 1, and this is what they like to ask about in 3rd and 2nd years. Okay, so even that this scenario is not with you, okay, you need to orient yourself with it, and you need to start having that analysis. So I'm going to quickly explain it, because this is not with us in the lecture. So here, this is a patient with diabetes insipidus, and this is a type of diabetes in which the ADH doesn't work. So let's recall, ADH, or vasopressin, is a hormone that is released by the pituitary gland, or the posterior hypophysis. And I'm sorry, let me just 1 second. Okay, I'm sorry. So usually ADH is a protein that is released by the pituitary gland, and what it does, it binds to the collecting duct in the kidney, restoring all the water that was about to be secreted into urine. So if you had hypotension, you'd want to secrete ADH to gather as much water as you can. So patients with diabetes insipidus, this ADH binding with its receptor is not working, either because the ADH is defective, or because the receptor, it's usually the receptor that's defective. Okay, so in that case, what happens is you get a lot of urine, a lot of water in the urine. So that explains his symptoms: increased thirst, frequent urination, nocturia, which is waking up to urinate, and it explains the osmolarity. So osmolarity is the amount of salts in urine, or the concentration of salts, and it's less than 300, which means that it's highly diluted due to the extra amount of water. So in that case, it's highly diluted. That explains his symptoms. So how is this related to protein trafficking? The doctor just wants you to know the receptor involved in this disease is not released. So there's a problem with protein trafficking. So he mentioned this scenario, and it's better that you start orienting yourselves with these scenarios, because even though they're not coming, like he won't ask you anything about diabetes insipidus, they can still like, although they might ask you, what's the hormone what's the problem in diabetes insipidus? It's just a mutated protein that is degraded, but I don't think they will. Okay, just keep it in the back of your head. So don't get scared when you see this. Just hi. Okay, so usually you have an endoplasmic reticular system. So in this system, when a ribosome is translating a mRNA, it gets into the rough endoplasmic reticulum, which then gets into the cis-Golgi. So the first part, this is the first thing that I want you to know from the entire lecture. The first part of the Golgi apparatus is called cis-Golgi. The latent part is called TransGolgi. So secretory vesicles, or these, are released from the TransGolgi. Is that okay? I think it's pretty simple. We can move on. Okay, let's start. So the general premise of it, you have an mRNA that gets translated into a polypeptide chain. Now, one of the questions that actually came in our final was that this part of the polypeptide, is it the N-terminal or is it the carboxyl terminal? Now, as you know, a polypeptide chain has a N-terminal and a carboxyl terminal. So let me just see your answer to see if you get it right or not. N-terminal or carboxyl terminal? N-terminal. Okay, good. Okay. It wasn't this straightforward. It was, what part of it was, if you know if it's N-terminal or not. The second part of it was something else. Okay. So this is a very important distinction that you should come to. This part is the N-terminal. Okay. Now, this part has a sequence, or called a specific signal sequence. Okay. And this sequence usually determines where it goes. Now, there are two pathways. It can either do half of the transcription in the cytoplasm and then continue it in the endoplasmic reticulum or within the organelle, or it can finish the polypeptide chain and then the polypeptide chain does whatever it wants. It can go to whatever. So this is called code translational translocation. So as it's translated, it gets moved, and translation continues in the organelle, or it can finish post-translation translocation. Okay, so this is the first thing that you should keep in mind. I'll talk more about it. Okay, you have this. What I want you to know is nothing. I'll talk about it more here. Okay, type. Now let's talk about this. Okay, so the signal sequence or the specific signal sequence is usually found on the N-terminal. So this is the N-terminal. It has a sequence of amino acids. So it's a certain arrangement of amino acids. Now, for instance, one of the amino acids that you should, one of the amino acids signal sequence is proline, lysine, arginine, lysine, valine. Okay, when the cytoplasm sees this, it immediately carries it to the nucleus. So it imports the protein to the nucleus. Okay, طبعا 100% you don't need to memorize this and know the sequence, but what you should know hydrophobic signal sequences are usually involved in the transport to the endoplasmic reticulum. So hydrophobic, when the sequence is usually hydrophobic, it gets transported into the endoplasmic reticulum. When there are a lot of positively charged, it can go either to the mitochondria or the nucleus, and so on and so forth. Okay, طبعا plastid is the chloroplast, which is found in plants, so I don't want you to know this. The main thing is that hydrophobic signal sequences or sequence of amino acids usually get into the endoplasmic reticulum. Hydrophobic, if you guys don't know what hydrophobic is, which I hope you know what it is, it's no charge. Okay, so it's fat soluble. It hates water. It's not charged because water is charged and it likes things that are charged. Okay, so let's talk about the fate of the mRNA that has a signal sequence compatible with the endoplasmic reticulum. So you have an mRNA which gets translated. The first part that emerges is the signal sequence at the N-terminal. Okay. Now, طبعا, this is the only part where they mentioned that the first thing that comes out is the N-terminal, yet they asked about it. So that's why it's very important you understand things and not memorize them, because you can easily miss this out. So the signal sequence located at the N-terminal, okay, will be immediately realized by a signal sequence receptor. Okay, so a signal sequence points to its receptor. Okay, so it will get recognized by a signal sequence receptor, which will move it to something called a translocon, or they gave it another name, sec translocase, but stick to the first one. Keep the other one in mind, because you don't know how they can ask. Okay, so they move it to the endoplasmic reticulum. It sees the sequence. It says, oh, this is a hydrophobic sequence. I should go to the endoplasmic reticulum. So it goes to the endoplasmic reticulum, and what happens is it binds the SRB, or the sequence recognition pattern, okay, but with the signal sequence creates a complex. This complex, it has GTPase activity. Let's break this down. So there's something called ATP, there's something called GTP, or guanine triphosphate. Guanine triphosphate, so it's called GTPase, so it breaks down GTP. Okay, so this whole thing, SRB, signal sequence, together مع بعض, they have a GTPase activity. So they break down GTP at the endoplasmic reticulum into GDP, and thus this opens the translocon. This is how it opens. Okay, this is how it opens, and then this releases the SRB for it to get recycled and bind to another SRB or signal sequence. Sorry. Okay. Now once it's in, I'm gonna just go in details and then tell you what you should know. Once it's in, okay, the signal sequence will get cleaved. Okay, it will get cleaved by the signal peptidase. So what cleaves the signal sequence? Signal peptidase, and then transcription will continue. So this is called translation. co-translational translocation. Co-translational translocation. Okay. So the translation will continue within the endoplasmic reticulum. And then you end up with a protein in the endoplasmic reticulum. What should you know? Here's what you should know. The first thing is that the signal sequence is at the end terminal, and that translation is held or stopped when the entire signal sequence is released, which is hydrophobic. The signal sequence with the signal sequence receptor together, they have a GTPase activity, and they help open the translocon to allow the polypeptide in. Then the signal sequence is cleaved. You should know this, and then translation continues. Okay. طيب. One thing that I want you to know from now is that if you think memorizing these steps is خلاص, if you memorize them, you're good. Well, I want you to know that your entire batch has already memorized them. Okay, I'm 100% sure that your entire batch has already memorized them. So the way you stand out is by understanding and know how things happen or why things happen. Okay, so that is usually how students get A's. Okay, when they understand, because I'm just going to throw a quick example because it just crossed my mind. In MSK, first years, the thing they do is they tend to memorize the muscles and the nerves, and they think, no, خلاص, I can get an A in MSK. Thing is, that's not how it happens. Okay. The reason is they usually ask about the clinical part of MSK. Okay. So you need to start understanding medicine. That's the point I'm trying to give to you. Okay. Even though it's mostly, it's memorization, you need to start trying to understand it at least. Okay. So that's that. So that's how the transport into the endoplasmic reticulum works. Now, once it's in the endoplasmic reticulum, okay, it can go to the Golgi. It can stay in the endoplasmic reticulum. It can go into the cis-Golgi. It can then, from the Golgi, it can either stay in as lysosomes, or it can go outside through secretory vesicles, or it can form a secretory granule. So it has different fates that you need to be oriented with. Right. Now let's talk about what if we want to transport it into the nucleus. Simple. So you have.
Ibrahim, I have a question before we keep going. Can you explain the difference between transcriptional and what is it called? Co and post translational.
So I want you to know that the transport to the rough endoplasmic reticulum is co. So as it's getting transcribed, okay, actually it's not as, it's the transcription halts or stops and then continues in the rough endoplasmic reticulum. But for instance, when an enzyme wants to get into the peroxisome, okay, the whole polypeptide is formed, it's done, خلاص, it leaves the ribosome, and then the recognition sequence is recognized by the peroxisome. Okay, so after translation, while in the rough endoplasmic reticulum, it's a co-translation translocation. Okay, makes sense?
Yeah, thank you.
All right. So you should know, endoplasmic reticulum is a co-translation. So it stops translation when it binds to the SRP or the signal sequence receptor pattern, or the receptor of the signal sequence. And then it continues, it continues in the rough endoplasmic reticulum. Okay, this is not the case, for instance, for proteins that want to get into the nucleus. So this is post-translational translocation. Okay, so here what happens is you have a sequence of polypeptides after its translation, and these polypeptides have something called a nuclear localization signal, or it's the same thing as this is specific signal sequence. They just called it nuclear localization signal, or NLS. Okay, why? I don't know. They just called it that. Okay, or you can call it 100%. It's valid to call it the signal sequence. Okay, so the nuclear localization signal binds to importin, or as it's called, the nuclear import receptor. Now this causes its transfer from the cytosol to the nucleus. Now, once it's in the nucleus has a lot of this protein called RAN-GTP. RAN-GTP will bind to the to this complex of the polypeptide and the important, and it will separate them. Then RAN-GTP will become RAN-GDP, okay, and bind to important. No, wait, what? Okay, I'm sorry, let's repeat this. So you have a polypeptide chain which binds to, which is recognized by the important, which gets into the nucleus. Now RAN-GTP binds to this complex, leaving the polypeptide outside or inside the nucleus, okay? Now, RAN-GTP and important will get out of the cell. Outside the cell, due to different conditions, RAN-GTP will become RAN-GDP, releasing or recycling important, and then it will get out and get phosphorylated in the nucleus, okay? But what I want you to know is that important is involved in the transfer inside, RAN-GTP is involved in the transport of important outside, and that unlike the rough endoplasmic reticulum, nothing cleaves the signal sequence. So the signal sequence is part of the protein structure. Does that make sense? Okay, perfect. I can go over them quickly at the end if you want, but let's try to understand as much as possible. Now, let's say you want to transport it into the mitochondria. It's even simpler. So what you want to do is the signal sequence is different for the mitochondria. It's going to get recognized by, first of all, chaperone proteins, which is HSP70. Now, chaperone proteins are proteins which rearrange proteins, okay? So when you have a polypeptide chain, it usually folds into a structure. Now, this folded structure cannot get into the mitochondria. So you need something that makes it a polypeptide chain again, okay? And this something is called a chaperone because it changes protein, and it's called HSP70, okay? So HSP70 unfolds the protein and allows for its transport into the mitochondria. So you have a translocase on the outer membrane, TOM, which binds to the signal sequence, letting it in, okay? And then with the HSP allows the protein in, then the signal sequence is cleaved by mitochondrial processing peptidase. So it's cleaved just like in the rough endoplasmic reticulum. And then it gets inside the cell through TIM, or translocase inner membrane. And once it's in the mitochondria, the other chaperone, which is another HSP70 protein, will fold it again to its original shape. Okay, that's the general gist of it. But what they can ask about, added to the list, here is the role of ATP hydrolysis. So ATP hydrolysis is used twice here. First of all, when the HSP70 dissociates from the polypeptide chain, okay, so that's the first place. The other place is when the HSP70 binds to the polypeptide, pulling it from the TIM, okay? So two places. And they can ask about how potential difference or the membrane potential is involved and in the translocation of mitochondrial proteins. And it does this by driving the protein into the inner membrane. So the potential difference is a factor that plays a role in transporting the protein into the inner membrane. Just keep that in mind, know where ATP is hydrolyzed. And yeah, that's all you should know from here. Okay. Now, the mannose 6-phosphate pathway. Like, previously in your presentation, they mentioned eye-cell disease, and then they talked about the mannose 6-phosphate pathway, but I don't want to do that because I think this is a more convenient way of doing it. So let's say you have a polypeptide chain. This polypeptide chain had a signal sequence to transport it into the, so it's usually in the cytoplasm, as you can see here, and it had a signal sequence to transport it to the rough endoplasmic reticulum. Now it's in the rough endoplasmic reticulum. The signal sequence is cleaved. Now it gets into the rough endoplasmic reticulum, then into the cis-Golgi, but it has a sequence which binds to mannose phosphate, okay, mannose 6-phosphate. Now, due to its sequence that binds to mannose 6-phosphate, okay, not all polypeptides are the same, but some have a certain sequence, okay, that they didn't talk about here, but that is able to bind to mannose 6-phosphate. Now, this mannose 6-phosphate, along with the protein, so you have a polypeptide with the mannose 6-phosphate, okay, this entire thing will get the TransGolgi. It will get recognized by mannose 6-phosphate receptors. And once it's recognized by the mannose 6-phosphate receptor, or M6PR, okay, this receptor indicates that it will be forming a lysosome. Once this polypeptide with the receptor binds, it gets cleaved into an early endosome. This early endosome has a channel for that makes it acidic. So the inside of it is acidic, as they mentioned here, with a pH of 5.5. Okay, so it's acidic. This dissociates the polypeptide chain and the mannose 6-phosphate. Okay, the polypeptide chain and the mannose 6-phosphate, when they dissociate from the receptor, the phosphate group is cleaved. And when it's cleaved, this allows the polypeptide chain to stay away from the receptor, so it doesn't bind again. Now you have the polypeptide chain in the late endosome, which is an enzyme. It's usually lysozyme, and it can form a lysosome. Okay. I'm sorry if this got confusing. Let me repeat it. So you have a polypeptide chain. It went through the entire sequence of the rough endoplasmic reticulum, or the entire process. But it also had a sequence which triggered it binding to the mannose 6-phosphate. Okay. The mannose 6-phosphate with the polypeptide will get recognized by the mannose 6-phosphate receptor. This allows it to be transferred into an early endosome. And this early endosome has a low pH, dissociating the mannose 6-phosphate and the polypeptide. This is one entity from the receptor, the second entity. So now you have mannose 6-phosphate with a polypeptide, which will, this late endosome will have phosphatase enzymes, which will break down the phosphate, leaving the mannose and the polypeptide. Okay, this mannose with the polypeptide are no longer able to bind to the receptor. This allows for the receptor to be recycled and the enzyme to stay in the lysosome. Okay, I hope this makes sense. This is what you should know, that mannose 6-phosphate is involved in the transport of the enzyme or the polypeptide chain to the lysosome. Okay. Now, anything that doesn't allow this to happen will cause a disease. Okay. So one of the mechanisms in which a disease can happen is the mutation of this enzyme, phosphotransferase enzyme. So Ibrahim, you didn't mention this enzyme. When did we talk about this? Okay, I'll talk about it. So the phosphotransferase enzyme is an enzyme which is needed to transport the mannose phosphate into the polypeptide chain on here. Okay, if this enzyme is mutated, the polypeptide chain, which was designated to go to the lysosome, will no longer go to the lysosome. So you will end up with lysosomes without enzymes. Now, lysosomes usually are found in phagocytes, or they're usually are used to degrade the organelles. So usually organelles such as the mitochondria, when the cell ages, will get into the lysosome. But if there are no enzymes to digest it or break it down, you end up with chemicals. This is called a storage disease. Okay, this buildup of chemicals is extremely bad for the body, and it causes widespread symptoms because it's a genetic disease. Okay, let's talk about it, and it's called eye cell diseases. So eye cell diseases, let's talk about it. They're caused by the mutation of phosphotransferase enzyme. When the phosphotransferase enzyme is mutated, it will no longer bind mannose 6-phosphate with the polypeptide chain. This causes lysosomes without enzymes, buildup of mucolipids usually, or buildup of chemicals. You don't have to know it. And this causes a lot of diseases. They love to ask about clinical significance. So they will ask you, what's the mutation in eye cell diseases? Or they will ask you, why doesn't eye cell diseases, why in eye cell diseases, the protein isn't transported. So you mentioned mannose-6-phosphate, it's no longer binding to the polypeptide chain, and hence you end up with a storage disease. Now here they didn't mention any unique symptoms that you should memorize, but you should know the enzyme mutation, and you should know that it's related to mannose-6-phosphate. So mannose-6-phosphate is needed to form enzymes in the lysosome. That's it for that pathway. Now, the other and last place in which an enzyme can go, or a polypeptide can go, it's into a peroxisome. Now, a peroxisome, as you should know, it's needed for lipid degradation. Okay, so anything that doesn't allow for this enzyme to enter the peroxisome, you end up with another storage disease. So lipids build up in the peroxisome, and this disease is called Zellweger syndrome. So what's the difference between Zellweger syndrome and the eye cell diseases? Before that, let's talk about the mechanism of transport of polypeptide into peroxisome. It's extremely simple. You have a polypeptide chain, post-translational translocation, with a sequence. However, this sequence is found on the C terminal. Unlike others, this one is on the C terminal. Okay, this sequence is recognized by peroxin, which is a protein found on the peroxisome. Once it binds to it, it enters. That's it. That's all you should know. Okay, and this signal sequence is made of serine, lysine, and leucine. But do I have to memorize this? Usually, when it's this simple, they can ask about it, but yeah, memorize it. Okay, memorize this. It's serine, lysine, and leucine. Okay, and it's found at the C terminal. It binds to peroxin, enters the cell. A mutation in peroxin will not allow the enzyme to enter the cell. Okay, so you end up with peroxisomes without enzymes. This is called Zellweger disease. So in Zellweger disease, you have mutations of the peroxin. In eye cell diseases, you have mutation of the phosphotransferase. So mannose-6-phosphate cannot bind to the polypeptide chain, and hence you don't get enzymes in lysosomes. Type. One last thing. Wait. Okay. We have two more things to talk about. Okay, and they're relatively important. I assume from the lecture of amino acids. Now that lecture might sound hard to tackle, but what I want you to know is that each amino acid has certain characteristics. Some are secondary amino acids. Some can form O bonds. Some can form sulfur bonds. So this is what you should memorize from it. Each amino acid with its specific sequence. So, and they're not. You don't have to memorize all the 20, but just 10 of them which have specific sorry which have specific sequences, specific characteristics. Sorry. So why am I mentioning this is that let's say you had a polypeptide chain, and this polypeptide chain had a signal sequence to allow it to enter the rough endoplasmic reticulum. Now once this is cleaved, it goes into the rough endoplasmic reticulum. Okay, and it's made of proteins, of amino acids, sorry. Okay, now it can get glycosylated. Now different proteins have different prosthetic groups that are added to it, such as glucose, such as metals, to form different functions. One of these prosthetic groups is saccharides. Okay, and they are used to form proper folding, provide protection, or for signaling. So they're used to form the structure of the protein. Okay, and the way they are glycosylated, there are two ways actually. So you have O-glycosylation and N-glycosylation. O-glycosylation occurs in the cis-Golgi. Okay, N-glycosylation occurs in the rough endoplasmic reticulum. So that's the first point that you should know. N-glycosylation occurs in the rough endoplasmic reticulum. O-glycosylation occurs in the Golgi. The way I used to memorize this when I was in first year is that Golgi. This O is O linked, and this is how I used to memorize it. It came in our final, it helped me. Like, the other thing which you can memorize now or memorize from the amino acid lecture is that the O-glycosylation occurs when there is serine or threonine. So from the amino acid lecture, you should know that serine and threonine are capable of forming O bonds, okay, or oxygen-linked bonds. Or you can just memorize it from now that when there's a polypeptide chain with serine or threonine, okay, they can get glycosylated by an O bond, which we said occurs in the Golgi. That's it. Okay. The other part which you need to memorize is that sometimes due to asparagine, which is amino acid which can form N bonds or N-linked, it can join the saccharide with the polypeptide through an N bond, and we said it occurs in the rough endoplasmic reticulum, is when you have asparagine. Okay. So that's the second point. Now you know where each occurs. Now you know which amino acids are involved in which type of glycosylation. Now let's talk about the last part, which is which saccharide is added to what amino acid. So in case of O-linked saccharides, you have N-acetyl galactosamine, and in case of N-linked, you have N-acetyl glucosamine. Okay, so glucosamine usually binds to asparagine, and galactosamine usually binds to threonine or serine through hydroxyl bonds. And why is this finding important? To form different structures of proteins, and this can be used as receptors or it can be used as different things. Now, one low yield point that they mentioned, but keep it in the back of your head, is that polypeptides which get N-glycosylated usually end with a negatively charged sialic acid. Okay, I don't think you should know this here because there is a lecture on it. I forgot what it's called. I think it was called carbohydrates, but there's a lecture on it that explains the structure of glycoproteins in way more details. Okay, so we've talked about three things. First of all, the location. Okay, where does each occur? You should know this. The amino acids involved in each bond. You should know this. The other thing that you should know is what saccharide is involved in what bond. And the last thing they mentioned, this low yield point, is that the N-glycosylated polypeptide usually ends with a negative. So you have this polypeptide. The first thing we said is glucosamine, which is a saccharide. So this is glucosamine. This is asparagine. This is N-link. It usually forms like a lot of saccharides, but it ends with sialic acid, okay, which is negative. Okay, there's a lecture on it. You can memorize it now or keep it till that lecture comes. The last thing we're going to talk about, and this is more of a neuro block. I don't know why they mentioned it here, but let's talk about it. It's extremely simple. Okay, just focus with me. So you have proteins which are involved in the exocytosis of some other hormones or polypeptides. So these proteins are syntaxin, SNAP25, synaptoprevin, synaptotagmin. Okay, what I want you to know is that syntaxin, SNAP25, and synaptoprevin, they usually bind each other when the vesicle is closed. Okay, and then you have a calcium channel, which is a voltage-gated calcium channel. When the action potential comes, calcium will enter. Calcium will bind synaptotagmin, and once it binds synaptotagmin will bind to this complex of proteins, allowing for the two membranes to come very close to each other. Once they're close to each other, fusion occurs and they release the hormones or the contents inside them. But what should I know? I think the only point that you should know is that synaptotagmin binds to calcium and it causes the release of the proteins. If you know that, that's more than enough. And one thing that you should keep in mind is the fact that some toxins or Botox, if you guys are familiar with it, some toxins bind to these different proteins at the cell membrane and cut them. So you end up with what? You end up with no exocytosis. When this happens, you don't get the release of the neurotransmitter here. Okay, so I'll give you an example. For muscle contraction to occur, you need to release acetylcholine. But when you give botulinum toxin, okay, you don't get the release of acetylcholine, so the muscle remains paralyzed. You can't contract it anymore. Okay, that's one example that you should know. Here's another example that when you usually read, you get scared, but I don't want you to get scared. I want you to read it, to get oriented with it, and not to memorize it. So let's just skip to the questions. What's the role of calcium? We said calcium binds to synaptotagmin, the yellow protein here, to allow the release of protein when we want it. What function of the neuromuscular junction is impaired? We said that acetylcholine acetylcholine will no longer come out of the vesicle, so you get paralysis, and this explains why he had weakness, muscle weakness. What else? طيب, how and the nerve conduction and is affected by the botulinum toxin, we said it. Which symptoms indicate that this toxin is affecting alpha motor neurons? You أكيد you don't have to know this because this is in the neuro block in second year. So skip it. Skip this. Just maybe answer these two questions, okay? Like, last thing they talked about constitutive secretion and regulated secretion. So usually when you form a polypeptide and you want to secrete it, you either secrete it as soon as you form it without regulation, that's constitutive secretion, or you regulate it. So you keep that polypeptide in the vesicle, such as the example we just talked about. You keep it in the vesicle, and once the stimulus comes or the action potential comes, you release it when you want it. That is called regulated secretion. يعطيكم العافية. Questions?
ممكن تعيد المانو 6 phosphate pathway please.
فكرت ممكن تعيد المحاضرة. طيب, مانوس مش راضي. لا. Just going to type. Let's share it again. Mannose 6-phosphate pathway. It's a very simple pathway. You have a polypeptide. It's we talked about how a polypeptide enters a rough endoplasmic reticulum. So you're done with this, okay? We have the polypeptide in the rough endoplasmic reticulum. What's next? This polypeptide, let's say this region, okay, has a sequence which is capable of binding mannose 6-phosphate using the enzyme called phosphotransferase, phosphotransferase, okay? When phosphotransferase joins mannose 6-phosphate to this sequence, okay, which is not found in all polypeptides, but if it's found, it will bind it. That means that this polypeptide is dedicated to go to the lysosome, okay? So this mannose 6-phosphate will get recognized by the mannose 6-phosphate receptor, which will cleave it and form an early endosome. I don't want you to focus on early or late endosome. Just know that it enters an endosome which has low pH. Okay, this low pH will separate the mannose 6-phosphate and the polypeptide, okay, away from the receptor. Now the receptor can go back to the cis-Golgi, and the mannose 6-phosphate will stay in the endosome. Now the phosphate group will be cleaved. What's the significance of the cleavage of the phosphate group? Is that now it's no longer mannose 6-phosphate, it's mannose, and mannose cannot bind the mannose 6-phosphate receptor, if that makes sense. Okay, so the polypeptide will stay in the lysosome. Anything that disrupts this whole thing, you will end up with eye cell disease. Does that make sense?
Yes, thank you.
All right. Any more questions?
What will they ask us about in this slide?
Okay, here are the questions that they can ask about. Thank you for reminding me. Usually here what they like to ask about is the fact, does translation occur before or after? So translation stops when the signal sequence comes out and it continues after. Okay, طبعا يعني standard, you should 100% memorize this entire thing. Your entire batch has already memorized it for sure. The extra points are that translation continues in the rough endoplasmic reticulum. That's one important point that you should keep in mind. Here, the point that no cleavage occurs, okay? You don't cleave the nuclear localization signal. I think that's an important point that you should recognize, along with this, okay? Here, I think the role of ATP and chaperone proteins, because I remember a question asked about it. Where does ATP hydrolysis occur? If I'm not mistaken, here you should 100% know eye cell diseases and phosphotransferases, and how if you don't have phosphotransferase, why do you end up with eye cell diseases? You should know Zellweger syndrome is due to a mutation in peroxisome. You should know the three things that I talked about: location, amino acids involved, and the saccharide that's involved, along with this tiny bit of information. And here you should know that synaptotagmin binds to calcium, and it's part of being regulated secretion. What else? That's it. That's all the questions they can ask. 2 of them are coming. Okay. So don't
Okay, thank you.
100% take my word for it, but these are all the high yield points that you should 100% know. Okay. What else? Any more questions?
Can you repeat the saccharides that are involved in the O and N linked?
It's simple. So you have 2 saccharides. You have N-acetyl glucosamine and N-acetyl galactosamine. N-acetyl glucosamine binds to وينها asparagine بس إذا لقيتها يعني وينها okay binds to asparagine okay glucose asparagine. N-acetyl galactosamine can bind to either threonine or serine because they're able to form O bonds.
Thank you.
Of course. طبعا if you have any questions feel free to feel free to send me an email or text me on WhatsApp or on Twitter or whatever you want. Okay. Yalla, I wish you good luck in your exam and بس if you need anything text me. وانا wish me good luck in my exam on Sunday.
Good luck.
سلام.